But I thought I'd start off by just introducing the topic of ubiquitin and going over some of the information, general introduction into ubiquitin and what it means before I get to some of the data. So ubiquitin is a highly conserved protein. It's 76 amino acids long. It's a highly conserved protein. There are only 3 amino acids that are different between the human and yeast orthola. And its classical function has been linked to proteasomal degradation. However, recently it has been linked to non-proteasomal function in various processes. I'll talk a little bit about that in a second. Can you see the mouse when I move it, Laura? Or, so.
Yes, Adriano?
You can
see the mouse.
Okay,
yes,
so I don't want
to. Good idea. Thank you.
So it's a globular protein. It has a hydrophobic globular core, and the amino terminus is here, and the carboxy terminus is here, and the carboxy terminus is the point of attachment protein. So ubiquitin classically is attached to substrate proteins through an enzymatic cascade involving 3 enzymes, an E1, an E2, and an E3. The E1 is called a ubiquitin activating enzyme. It requires some energy to adenylate or activate ubiquitin, whereby it forms a thiol ester intermediate with an active site cysteine residue. The E1 interacts with the E2, and ubiquitin is actually transferred from the E1 to an active site cysteine residue on the E2. And the E2-E3 complex can form a structure known as an E3 a ligase that has the ability now, in this case it's prime, that has the ability to transfer that ubiquitin molecule to a target protein. And in this slide here, the green structure here represents a substrate protein that interacts with the E3 ubiquitin ligase. So the substrate protein has a lysine residue, and it's the epsilon amine group on a lysine side chain on the lysine residue that gets attached to ubiquitin. So once the target or the substrate is bound to the E3 has a recognition sequence that recognizes what is known as a degradation signal in the target protein, or essentially a binding motif within the substrate protein that binds to the E3. And once this interaction occurs, then the E2 is in close proximity. It can transfer the associated ubiquitin to the epsilon amine group on the lysine residue of the substrate protein. And this is a covalent interaction. And in some cases, so it's a covalent interaction. It's an isopeptide bond because it's formed between carboxy terminus ubiquitin and epsilon amine group of a lysine. So it's not a true peptide bond. But the ubiquitin that's attached can also be attached with additional ubiquitin molecules. Ubiquitin can be attached to itself to give rise to these polyubiquitin chains. Typically, it's been thought that polyubiquitination of proteins targets them for proteasomal degradation. So a protein would be multi-ubiquitinated, and this would then be recognized by the proteasome, and the proteasome would be able to degrade the substrate. Meanwhile, ubiquitin would be removed by deubiquitinating enzymes and recycled. Ubiquitin is a very interesting molecule, and as I said here, it can form polyubiquitin chains. It has 7 internal lysine residues. So chains can form through conjugation to any of the internal lysine residues. Notable residues are 48K63. So polyubiquitin chains can be formed through lysine 63 or lysine 48. In fact, they can be formed through modification of all of the lysine residues. The most studied have been K63 and K48 link chains. I'm going to talk about them because we've looked at binding of our ubiquitin binding them into polyubiquitin chains, K48 and K63. Laura, can you hear me okay? So as I said, you could have polyubiquitin chains that are either formed through lysine 6, or which is, so here are the 7 internal lysine residues, 6, 11, 27, 29, 33, 48, that are in ubiquitin, and lysine chains can be formed, as I said earlier, through polyubiquitination of that particular lysine to give rise to what are known as homotypic polyubiquitin chains.
Adriano, we can hear you just fine.
Okay,
No problem.
okay, great, thanks. And then we have potentially mixed chains where you might have branch chains formed through one particular, like a K48 type chain, and a branch chain formed through a K29 linked chain. Or alternatively, you could have, this is the substrate protein, or for example, and you could have multiple mono-ubiquitination events on separate lysine residues to give rise to something known as multi-mono-ubiquitination. Now, the reason why I talk about this is because it's important. Different types of chains appear to have different functions. So, typically, K48 linked chains have been linked to proteasomal degradation. So if a protein is modified by K48, so it's been thought that that would then target that protein for proteasomal degradation, whereas modification by other types of ubiquitin chains, such as K63, would serve non-proteasomal functions, such as in DNA repair, endocytosis, and potentially activation of protein kinases, among other types of functions. And this is interesting to us because K63 has been linked to endocytosis, and that's part of what we study in my lab. And mono-ubiquitination itself has been linked to endocytosis and endosomal sorting and other functions, and multi-mono, so different lysine residues being modified by a single molecule, have been shown to be important for endocytosis. So, as I said earlier, part of some of the types of ubiquitin linkages target proteins for proteasomal degradation. Now, the mechanism for that is thought to, well, just before I get into that, so polyubiquitin chains can target proteasomal degradation, or alternatively, modification of receptors on the surface of the cell. And this is a receptor we study in my lab called SPR4, the G protein coupled receptor, chemokine receptor that's implicated in various diseases such as cancer and HIV infection. Modification of this receptor by ubiquitin, in this case the E3 ubiquitin ligase, AIP4, targets the protein for lysosomal degradation. There's a machinery that recognizes the ubiquitin moiety, and that machinery has what are known as Ubiquitin-Binding domains, and I'll talk a bit about them in a second. But the principle behind the modification and the function is linked to what are known as Ubiquitin-Binding domains, or UBDs. So the proteasome, for example, has what are known as ubiquitin receptors, and these are the binding domains that actually recognize the ubiquitin moieties on the substrate, bind to them, and sequester the tag protein for ultimate degradation through the proteasome. Alternatively, there might be an adapter molecule that has a ubiquitin binding domain, so it could be an indirect interaction of a substrate with the proteasome. Nevertheless, the ubiquitin binding domain is served here as a recognition element to target the proteasome. And the same thing exists in endosomal sorting. So this is this would be a very complex figure, and certainly the take-home message here is just to let you know that there's a process by which proteins such as cargo that are modified by ubiquitin. This cartoon here represents a receptor that's been modified with two ubiquitin moieties. And the whole point here is that the machinery that sorts or targets proteins for lysosomal degradation on endosomes has what are known as ubiquitin binding domains, and there's 4 different protein complexes, 3 of which have ubiquitin binding domains, so they're able to recognize ubiquitinated cargo. And since research in my lab is on understanding how proteins are downregulated in this or downregulated by lysosomes or targeted to lysosomal degradation, we're very much interested in identifying novel ubiquitin binding domains that might play a role in this process, in addition to the ones that have already been discovered. So, as I said earlier, ubiquitin is a globular protein. This is a schematic ribbon diagram and just showing here in D here the lysine residues that are modified by ubiquitin. All of them are exposed and all of them can be attached if ubiquitin chains to itself. K48 and 63 have been the most widely studied. Ubiquitin binds to most ubiquitin binding domains through non-covalent interaction. So the interactions with UBDs are non-covalent through a hydrophobic patch, typically centered around isoleucine 44. Other binding domains have been linked to aspartic acid 58 on ubiquitin and also interactions through glycine 76. I think one example exists for that. So isoleucine 44 forms a nice patch that interacts with multiple ubiquitin binding domains. And I think there are probably about, last count, 22 known ubiquitin binding domains. And most of them interact with lysine 44 on the ubiquitin moiety. And this is just a three-dimensional ribbon diagram of examples of ubiquitin binding domains. Typically, they're also helical in nature, and they mediate interactions through the helicine with a patch of isoleucine 44 and leucine 8, which also, and they can be in various sizes, small, 20 amino acids long, or bigger actual protein complexes. But they all typically interact with isoleucine 44, which is kind of interesting. Now, I talked about K48 link chains and K63 link chains. They're recognized by different ubiquitin binding domains typically, and part of that has to do with the structure. So K48 link chains are very compact. So here's just an example of two ubiquitin moieties that are linked by lysine K48. And it's a very compact structure, whereas the K63 link chains are more extended. There's a bit of a separation between the ubiquitin moieties, and this dictates binding preference for various ubiquitin binding domains. So different UBDs would have different affinities, K48, K63 link chain, typically, not always typically. Now, we compared our ubiquitin binding domain with these types of chains. Now I'll get into that in a second. Might have hit a bit of a roadblock here. I'm trying to advance my slide, and it isn't moving. Sorry about that.
Adriano, is it working okay?
I'm trying to advance it. It isn't advancing. Let's see.
How about if you go back?
Okay.
Does it go back and then forward?
I got it. It's going now.
Okay. Oh, good. Excellent.
This is just a three-dimensional representation of ubiquitin, and this is K48, and this is the proximal ubiquitin and the distal ubiquitin. And here's what is known as the ubiquitin binding domain. It's called the UBA type domain, and it prefers K48 link chains, partly because of the orientation of the two ubiquitin molecules within the structure of the UBA. You can see where it fits into this nice little pocket here that's formed by the two the proximal and distal ubiquitin moiety. This is just an example to show you why perhaps a K63 link chain wouldn't be able to bind to this type of ubiquitin binding domain. So we're very much interested in understanding how ubiquitin binds to our protein of interest. And what we've done is initially we've characterized these interactions by simple GST pull-down assay. So this particular plot is showing an example of the protein that we're looking at. And this is a typical in vitro ubiquitin binding assay. our protein is expressed in cells, and we can take cell lysates and incubate them with either GST ubiquitin that's fused to GST, or the mutant ubiquitin, in which leucine 8 and isoleucine 44 have been changed to alanines. As I said earlier, 8 and 44 form the major binding site for most ubiquitin binding domains. What we find is that we can detect our protein in this type of interaction very nicely. So in a pull down, what's interesting is that we do not detect our protein when our cell lysates are incubated with ubiquitin. The mutant form of ubiquitin that doesn't bind to most ubiquitin binding domains. So we've defined what we consider to be a ubiquitin binding domain that has a preference that is recognized by the typical motifs on ubiquitin for a non-covalent interaction. So this just shows the level of expression of the fusion protein that we use in this type of assay. We could also define whether or not our ubiquitin binding domain binds to K48 or K63 link chains by using these GST pull-down assays. So here are purified fusion proteins, and we have defined at least 4 ubiquitin binding domains. I've called them 1 to 4 here, UBD 1, 2, 3, and 4. And we've incubated them with either K48 linked chains, and these are tetra ubiquitins, so 4 ubiquitin molecules that are linked through K48 or K63. And what we find, again, this is a simple pull-down assay, incubated for about 1 hour, and then eluted proteins are resolved by SPR page and immunoblotted for ubiquitin. What we can see here is that K48 link chain binds very nicely to our ubiquitin binding domain, except UBD2 does not recognize K48 for some reason, whereas K63 also binds very strongly to our ubiquitin binding domain. And of course GST does not. The binding is very strong. Ubiquitin binding domain 3 seems to prefer K63 better than the other 3 ubiquitin binding domains, and it seems to bind to 48 as well, very strongly. We can compare the intensities of these signals between plots and through graphs, but it's very difficult to determine whether or not our using this type of assay, whether or not our ubiquitin binding domain prefers 48 or 63. It does suggest that perhaps it prefers K63 versus 48 just because the intensity of the signals are a little bit stronger. These particular blots were taken from the same gel, so the exposures are similar, although our input here exposure is a little bit dark. So it's tough to tell. And what I'm getting at is that this type of assay is good to tell you that you have perhaps ubiquitin binding domain, but you need a more sensitive type of assay to actually start getting at preferences, an assay that would be able to give you affinity. We've also done other types of ubiquitin, in vitro ubiquitin binding assays using purified or immunoprecipitated protein. So we would immunoprecipitate either a wild-type protein or a mutant version of the protein in which the ubiquitin binding domain has been mutated. And in this type of assay, when we immunoprecipitate our protein here, either the wild-type or the mutant, shown here in the lower panels, and incubate with K48, we don't see any K48 binding in this type of assay. Whereas the K63 binds very efficiently binds somewhat efficiently, I should say, to our immunoprecipitated protein, and not to the mutant. So this has led us to conclude that perhaps our protein has a ubiquitin binding domain that prefers K63 link chain versus K48 link chain. But it's also difficult to predict just based on this type of assay. We need a more sensitive assay. And this brings me to SPR, which is something that I'm very interested in doing because these ubiquitin binding domains are nicely characterized, these plasmon resonance, which is I'm just going to go over it just very briefly. SPR is a collective electron oscillation happen at at a metal dielectric interface, senses the refractive index change within a thin layer on the surface. And this change takes into consideration the mass of the protein, and one can predict the potential refractive index caused by an interaction with one protein to another that's been immobilized on a metal surface. So it can detect these changes on the surface of the chip, and the advantage obviously is that you can get high resolution kinetic analysis using this feature, which is exactly what we want. We know that we have these Ubiquitin-Binding domains. We don't know what the affinities are. We don't know the relative affinities and the preference K48, and that's important for us. Knowing that could allow us to decipher function eventually. The SPR system consists of fluidics, optics, sensor chips, controllers, and software, which is relatively easy to use. We've been trained on it. My technician knows how to use it quite easily. So it's a very easy interface and very easy to design experimental steps and then analyze sensograms. So typically you'd have two molecules. One molecule would be the ligand. The ligand would be immobilized on the surface of the chip, and then we'd have the analyte, which would be flowed over, injected, and then captured perhaps by binding to the ligand, which is immobilized on the surface. So in our case, what we've done, and I'll talk a bit about it in a second, but just to briefly say, is that we've immobilized our Ubiquitin-Binding domain, and we've injected various concentrations of Ubiquitin to get those response curves. So the ligand would be our Ubiquitin-Binding domain immobilized, and the analyte would be various concentrations of our Ubiquitin that are flowed over. And of course, as I said earlier, it's very useful for determining typical basic kinetic principles of the interaction. One can obtain the association constant, disassociation constant, and using this information to calculate the KD or the affinity of the interaction. So this would be a typical sensogram where the analyte would be injected, and the refractive index would be, the response units would be measured by taking the refractive index, which would be shown by these curves here, and differences in refractive index are easily distinguished between various concentrations of ligand. So for us, the type of interaction that we're looking at is a protein-protein interaction, and actually our Ubiquitin-Binding domain is a peptide. So we've been able to make a peptide of our Ubiquitin-Binding domain and then flow Ubiquitin. It's worked really well for us. We've tried different approaches. And again, this is just a quick overview of the optics. So polarized light would be refracted at a different index dependent upon the binding of one protein to the other, and then there'd be a detector, and this would be the sensor chip. So the flow would be in this direction, and then the detector can detect small changes in binding. So it's a very sensitive assay, which has worked to our advantage. And the other advantage that we've come across with this particular instrument, the Proteon SPR 36, which our department actually had, and we've been able to use quite extensively, is that you can immobilize 6 different surfaces. So with ligand, 6 different ligands or 6 different concentrations of the same ligand within one chip, and then flow over in a different, and this would be perhaps in the horizontal direction, and then flow 6 nanoites in the vertical direction. And I know I'm probably talking about that very quickly, but the advantage is that you could do multiple dose response curves with different ligands, which offers the ability to analyze to get full-blown dose response curves within one session on a single chip, which is what we've done with our study. And of course, that allows for, as I said, in one afternoon, you can get a full analysis and get good concrete data from a single chip. So what we've done is our ubiquitin binding domain is actually a biotinolated peptide. So some of these ubiquitin binding domains are actually pretty small. Some of them can be as small as 20 amino acids, and ours is very close to being 20 amino acids. So we've been able to actually synthesize it as a biotinolated peptide, which has worked to our advantage for using the SPR. So we in a typical experiment, what we would do, we would immobilize different amounts of the peptide in the 6 different channels. We would usually have 5 different concentrations, and then we would have a channel that left blank as a background control. And then in the vertical direction, and we could flow over our analyte, which would be our different concentrations of our ubiquitin. One of the advantages of the experiments that we've done is that we've been able to regenerate our chips. So we could, the ubiquitin UBD interaction is quite stable enough to measure in this type of assay, but it's also sensitive to high cell concentration. So we could remove down ubiquitin, regenerate the chip, and flow over additional ubiquitin concentrations. So in one sitting, we could do we were doing 12 different concentrations of ubiquitin to give us a really nice, elaborate dose-response relationship. Then you would generate sensorgrams from each of the 6 ligand channels of all the different concentrations of ubiquitin that you use. So, which is advantageous to us as well, because we can compare findings in the various channels that have different densities of our peptide. And another advantage of being able to regenerate the chip and be able to use it to flow over additional concentrations of ubiquitin is that we were looking at 3 different types of ubiquitin: mono-ubiquitin, K63 link chains, and K48 link chains. So in one sitting, we would be able to regenerate and flow over, I'd say, probably 9 different times ubiquitin molecules over the chip. So very, very efficient. So the type of chip that we used was a degradation chip. As I said, our peptide was a biotinolated peptide, so the degradation would bind the biotin. With the advantage of this for us was that the orientation of our peptide was always we would always know the orientation of our peptide. Typically it would be in this type of orientation where the biotin would be interacting with the degradation, and the peptide would be exposed to the lumen, if you will, of the flow channel. So we would flow over ubiquitin and binding reaction would occur, and this would be sensed and measured in this type of sensorgram. And of course we would have a blank, and we'd be able to track off our blank, our background very easily with the software that's available. And this is a typical sensorgram of our immobilized peptide. In this particular case, the concentration of peptide over was 0.1 micrograms per ml. So in terms of actual quantity of peptide, initially one of my fears was that we were going to be using a lot of peptide to be able to do these types of assays, and in fact, very small amounts. So you're allowed, the volumes that we're talking about that we were injecting were probably in the 200 to 300 microliter range. And we were using very minimal amounts of peptide, which is, in terms of expense of peptide, it was very minimal. And considering the amount of data that we would collect, it's very cost effective for us anyway. So this is a typical femtogram response unit. So our baseline would be set to 0 after background subtraction, and of course, once you inject the peptide at this point here, the response unit elevated. I think we measured from the time we stopped the injection was about 60 seconds. Typically, we were doing 60 seconds to 2 minutes. So the response units in this particular case reached a maximum of 60 units, which for us was more than efficient to be able to detect our interaction with Ubiquitin. So, as I said, we could flow over multiple concentrations of Ubiquitin. At any one time, we were just flowing over 5 concentrations, but in one sitting, we could regenerate the chip and flow over up to 15 concentrations if we wanted to. And I think we ended up doing as many as 12 different concentrations. And this is a typical femtogram after background subtraction. It looks pretty good. The higher the dose, the more binding you would get. And this will be reflected in the higher response unit. The higher response unit measured here. And of course, we could effectively get a nice dose-response relationship. I'll show you the data, how it looks when it's graphed. So this was mono-ubiquitin. And we also did load over a K63 like tetraubiquitin chain. This is a typical femtogram after background subtraction. We got very interesting looking femtograms. So, and here's I'm going back to the mono-ubiquitin. So upon injection, our interaction or refractive index would be almost instantaneous. And you'd see it level off, and then it would, as soon as we stop the injection, the ubiquitin would fall off, and the response unit turn close to basal. So the association is very rapid, and the disassociation in this particular case is also very rapid, although it's not completely disassociated. Again, our injection times are usually around 60 seconds. We were injecting, again, very small volume. And probably, I don't think we were using more than 200 to 300 microliters of our sample. In the highest concentration of ubiquitin, I think that we injected was 1,000 microliters. But it never returned in this particular example. The baseline never returns to 0, so the off rate is a little slow in these particular experiments. So we could remove the ubiquitin by treating our samples for regenerating to high cell concentration, which is very advantageous, but allowed us to use multiple concentrations in one sitting. K63, again, when we injected it, the higher concentration obviously gives the highest response, and we saw a very nice dose-response relationship. The shape of the curves are interesting in that there's a rapid, looks almost 5 phases in this, there's a rapid increase in our response unit, followed by a tapering off, and then it reaches somewhat of a maximal response. This association is a little bit slow with these guys. We're still trying to figure this out as well. So it didn't return to baseline immediately after our injection. And what we're trying to determine is exactly how ubiquitin is interacting with our ubiquitin binding domain. But anyway, we did get very nice specific binding to our peptide with K63 linked tetraubiquitin chain. We saw a similar pattern with K48 tetraubiquitin chain. Nice rapid increase in binding that sort of tapered off and then reached the maximum. Again, this association was somewhat slow. And as I said, we're still trying to come to grips with that. But we can do this over multiple experiments and generate very nice data. And this is an example of some of our experiments with mono-ubiquitin and with K48 and K63 ubiquitin, percent down, percent maximum down. So what we found is that mono-ubiquitin bound very nicely, specifically, with a very low affinity, though, compared to K63 and K48 link chains. We were unable to really reach maximal binding. We haven't been able to get a higher dose, so we haven't succeeded there yet with mono-ubiquitin. But with K63 and K48, we're starting to get to maximal binding. You can kind of see where the maximum binding is. In this particular example, it looks like K48 might be binding with a marginally higher affinity than K63. Probably our KDs are in the 1 to 3 micromolar range, which is very high, actually. It's a high affinity for doing SPRs type binding experiments, ubiquitin and ubiquitin binding domain. And this is typical of mono-ubiquitin. It's usually in the 500 micromolar range, these types of experiments. The K40 and K63 definitely bind with higher affinity for ubiquitin binding domain. We don't know if there's a difference, though, between the affinities as much as suggested here. Again, this is probably about 1 to 3 micromolar difference. Just a dose response of K63 and K48 from 2 channels that were on the same chip, but with biotinylated peptides plated at different densities. And the pattern is very, very similar in that we still get a rapid increase in binding. Again, we haven't, we're probably getting very close to maximal binding saturation. We just need to, we still need to do these experiments where we're using higher doses or higher concentrations of our tetraubiquitin. We just haven't been able to do that just yet. But graphing it and extrapolating, we can get high affinity binding in the micromolar, 1 micromolar range between K40, which show very, very similar affinity. And here's another example of a dose-response relationship with K63 and K48. And of course, these dose-response relationships that I'm showing you, response units, and all these different doses typically were done on the same chip that was regenerated, and we were able to inject multiple doses in one session, essentially, to get really full-blown dose-response relationships, which is very good. So within an afternoon, we can generate these very nice curves very easily, very reproducible. So just to summarize that, so I think we've identified a novel binding domain. You can characterize these ubiquitin binding domains by several techniques. And we use typical GSD pull-downs with ubiquitin for immunoprecipitation of our protein and incubating with purified ubiquitin chain and detecting binding just by immunoblotting. Obviously, that doesn't allow you to calculate kinetics of the interaction and the pharmacodynamic principles of the interaction, which is why we turn to SPR to define this ubiquitin binding domain, which suggests that it has a high affinity for K48 like polyubiquitin chain. Not sure that there's a difference between binding them the two. So we're still characterizing this interaction a little more, and obviously we're trying to determine the function of the ubiquitin binding domain. I think I'll stop there.